Hi, my name is Scott Edwards. I'm a professor at Harvard University. So what we're going to do is use genetic markers to study phylogeography a bit, but we're going to go beyond that and study aspects of the organism that tell us about the ways in which it interacts with its environment. Specifically, we're going to study gene expression and how that interacts with, in our case, a bacterial pathogen, a disease that's being inflicted on this bird that you see here, the house finch. This is an area of molecular ecology known as host-pathogen interactions. And these sorts of interactions are very exciting because they offer the possibility of looking at coevolution, the ways in which a pathogen, for example, might cause the host to evolve, which in turn might cause the pathogen to evolve yet again. So there's a really exciting bunch of questions that one can ask when one has a system involving a pathogen and a host. So, to introduce the two main characters in our story here, house finches are a very common species of bird in North America. You can find them from sea to shining sea, from all the way on the west coast where they're native to the east coast where they've been introduced in the 1940s. We're also going to talk about a bacterial pathogen or disease-causing organism called a mycoplasma. Mycoplasmas are very small, single-celled bacterium, and they're, in this case, very debilitating for the house finches, as we'll see. Here's a brief synopsis of what's happened between house finches and mycoplasma. In about 1994, scientists realized that house finches in the mid-Atlantic states were getting sick. They were getting huge amounts of swelling around the eyes. They were experiencing what we call conjunctivitis. They had trouble breathing. All of these were eventually traced to a pathogen which actually originated from poultry, from chickens and turkeys, and which is known as Mycoplasma gallisepticum. This is a very common pathogen of poultry, and in this case underwent a relatively uncommon jump from its normal host to a new host, the house finch. Now, the interesting thing about this scenario is that house finches appear to have undergone evolution in the short time after they got infected by this disease. 9 years later, biologists could detect that house finches were becoming resistant to the pathogen. They were showing less incidence of death. They were showing evidence of having encountered the disease but having survived. And they were showing a lot lower incidences of the swelling and the respiratory problems that I mentioned. So the main questions we're interested in are, how has the host, in our case the house finch, evolved in response to this novel pathogen that it had never seen before? Secondly, how has the parasite evolved? Since it's jumped from a host, the poultry, to a new host, the house finch, which is phylogenetically quite distant. These house finches and poultry haven't seen a common ancestor since about 90 million years ago. So it's not a trivial thing to jump from a chicken to a house finch. Here's a recent history of house finches. As I said, they are found in the western US naturally. They were introduced to the eastern US when the laws changed such that pet traders it was illegal to hold fancy birds as caged birds. So like good biologists, they simply opened their cages and let the birds free on Long Island. The result was that over the next 60 years, these birds spread all the way from New York City, clear across to the Rocky Mountains, where they're now abutting their former ancestors. Okay? In addition, house finches are found in Hawaii, where they were introduced presumably in the 19th century. When Joseph Grinnell visited the Hawaiian Islands in the early 1900s, he found house finches on every island, which suggested to him that they had been here for several decades. So his estimate was that they had arrived in the 1870s. Now I want you to remember these recent time frames because you'll see how the genetic data gives us a very high resolution picture of the differentiation of these populations when we use a lot of markers, even though the time frame has been very short. Okay, here's a movie showing the spread of mycoplasma from its beginning in the mid-Atlantic states all the way across the eastern US. It took a mere 5 years to move from a single point of origin all across the entire eastern US for this pathogen to spread. Extremely fast, but also extremely well documented because the Cornell Lab of Ornithology conducted what's called a citizen science survey, wherein bird watchers and people watching their bird feeders would report in on the internet whether they saw sick house finches. In this way, they were able to track the spread of the disease with unprecedented clarity. And it's turned out that citizen science is an extremely powerful way for biologists to collect ecological data, and you should look for an opportunity to become involved with a citizen science project. Now, we're going to look at 3 different time points in this study. The first is what we call the pre-epizootic stage in the 1980s before the pathogen arose. You can see that across the entire US and Hawaii, everything's blue, meaning there's no evidence of disease. Now, fast forward to 2001, and we can see we're in a situation where half of the country half of the birds in the eastern US have been hit by this pathogen, whereas the western US is free. Now, in the 1980s, that's a useful time to look at patterns of genetics because now the birds have been unaffected by the disease. By contrast, in 2001, we've got the eastern birds which have experienced this disease now for 5 or 6 years, whereas the western birds are still naive. Finally, we can look at a third time period in about 2007, wherein the disease has now jumped the Rocky Mountains, come down south through California, and is essentially free only from a few states in the Southwest. Here's a situation where we have Alabama populations which now have been with the disease for 7 years at least, whereas Arizona populations still have never seen the disease. As we'll see, these two populations have very different responses when they encounter the pathogen in an experimental setting. Okay, let's just review these time periods. We've got the pre-epizootic population based importantly on museum specimens that were collected by museum collectors in the 1980s. Museums and their collections have been a very important source of information from us because they have essentially provide a window into the past. In our case, into a time when the species had not seen the pathogen. We've got our 2001 phase. In this study, we're going to look at gene expression. We're going to look at how birds from Alabama respond to the pathogen when they've been exposed to it in an aviary setting. Okay? Finally, we're going to look at the 2007 situation, and we're going to again do a gene expression study, this time comparing birds from Alabama and from Arizona. Remember, Arizona still, at the time of the study, hadn't seen the pathogen, although we actually know now that it's beginning to sweep through Arizona, and we're ready to look at these birds as the disease is sweeping through. We're going to catch evolution in action. Okay, so let's look at the first study, a study of genetic variation. We're going to look at some of the same phylogeography principles that we've heard about in the first 2 lectures. Here we're going to use a type of genetic marker called AFLPs. This stands for amplified, fragment, length, polymorphism. Doesn't give us the detail of actual DNA sequences, but it allows us to look at genetic variation in terms of fragments. And what you can see on this gel is the huge diversity of fragments that we're getting from different individuals. Each column on this gel is a different individual. You can see the different populations that we've sampled. We've even sampled 2 related species to the house finch, the purple finch and the Cassin's finch. In this study, we looked at over 160 individuals from 16 different populations. Using the AFLP technique, we were able to look at 363 different markers. This is like having 300 different genes to look at. It's a very useful approach for getting large numbers of markers. It turns out that over half of these, over 60% of these markers were variable between populations, which gives us a lot of information. And you can see these variations directly on the gel if you look at the pattern of banding. Now, remember we talked about FST in the very first lecture. Here you see a pie chart showing a type of FST called AMOVA. What this is saying is that imagine the whole circle being all of the genetic variation within the entire species. Now imagine we're going to look within single populations, and we're going to just average the amount of variation that we find within each of the 16 populations. Turns out that we can find over 70% of the variation in the entire species when we look at just a single population. Okay? What that tells us is that house finches are not strongly structured. If you look at one population, you might as well have looked at them all. Okay? So 70% of the total genetic variation can be found within a single population. Nonetheless, we find a substantial amount of variation among different populations within each subspecies, about 8%. And when we look at yet different subspecies, so for example, the subspecies in the US versus the one in Mexico, we find over 20%. So there's detectable variation at different levels in the species, but within the US populations, it's a fairly uniform species genetically. Let's look at genetic diversity. This is very important to get a snapshot of this species before the disease struck. On the y-axis, you can see an estimate of nucleotide diversity. This is our friend theta or pi, what we've seen in the previous lectures. What you can see is that the amount of diversity in the introduced populations in the eastern US and Hawaii is pretty much the same as the amount of diversity in the native situation in California. What this tells us is that the eastern populations did not undergo a strong founder effect. They did not lose a lot of genetic variation. Now, using other markers, other researchers have found a small but detectable decrease in genetic variation. Personally, I think it's still, the jury is still out. We need to do yet further studies to definitively answer the question, has the eastern population lost genetic variation? This is important because it may be that the disease could spread faster in populations that don't have as much variation in their genes. Because of course variation is the stuff of adaptation. That's how populations respond to changing environments. Now remember how we looked at that time course and how the eastern population was founded only about 70 years ago and the Hawaiian ones in the late 19th century. Using these 363 markers, we're able nonetheless to pick out these birds with almost complete certainty, despite the fact that they've been separated for less than a century. So, for example, the western birds at the top cluster quite distinctly from the eastern birds in yellow and the Hawaiian birds in red. And this shows you the power, the extraordinary power when one uses a lot of genetic markers. If you showed this plot to a evolutionary biologist, they would tell you, oh, you're dealing with 3 different species. But of course, we know that these populations are really all part of the same species and having been separated only by several decades. Now, we can look at patterns of evolution in the phenotype of these birds in these 2 plots. And this is work done by my collaborator Jeff Hill at Auburn University in Alabama. What you can see is that on the left, that the epizootic actually differentially affected males as opposed to females. It turns out that the proportion of males in the population of Auburn, Alabama declined as the epizootic ensued. And this is interesting. Why might males be hit harder than females? Well, maybe males have more testosterone running through their systems, which might compromise their immune systems. There are lots of reasons why males might be hit harder by a disease. On the right side of the panel, you can see the way in which, within the males, redder birds were actually better survivors in this case than were dull birds. House finches come in a variety of colors, from bright red to orange to yellow. It turns out that the percentage of bright red birds in the population actually increased after the epizootic. This might mean that red in this species is an indicator of health and fitness. Maybe dull birds were differentially affected, negatively affected by this disease. Now, paradoxically, it turns out that smaller birds tended to survive the epizootic rather than larger birds. You can see how these indices of body size, we've got weight and tarsus length, all of the surviving populations were actually smaller than the populations before the epizootic. And of course, females were affected much less in general than were the males. Those are the bars in brown. But you can see how looking at different sections of the population gives us insight into how the population is changing as a result of this epizootic. All right, let's look at two studies looking at gene expression in the 2001 period and the 2007 period. We're going to use two different approaches. One, an older approach called Macroarray, in which we look at gene expression using nylon membranes. And then another called Microarray, where we look at gene expression using glass slides. Genomics is such a fast-moving field that even these approaches are being superseded by even more higher resolution approaches, which we're currently using. What we've done here is an experiment where we have taken birds from the wild, from Alabama, and infected them with this Mycoplasma pathogen, a particular isolate. Then we've asked, how have these birds responded to this infection by their gene expression? Each of these bars indicates a comparison of the gene expression of infected birds versus uninfected birds. So for example, you can see that in this orange slide, this orange bar showing HSP90, this is heat shock protein 90, you can see that the expression of that gene was higher in birds that had been infected by the disease as compared with birds that had not been infected. This tells us that HSP90 is one of the response genes to this pathogen. And you can actually see many genes of interest in this slide, all of which paint a portrait of the immune response of house finches to mycoplasma at the molecular level. Now, intriguingly, some genes which we thought would be upregulated, that would increase in expression because they're involved with the immune response, turns out they are actually downregulated when birds get infected. So for example, in blue here, you can see this MHC class II associated invariant chain, or CD74. That's a gene which is very important for the immune response. It actually went down in expression in infected birds. You can actually see that the MHC itself, MHC stands for major histocompatibility complex. It's also known as HLA in humans. Very important part of the immune system. MHC genes themselves are actually going down in expression when they're infected. This is a real head scratcher. Why would genes important for a response like this go down in expression? Well, it may be that this pathogen is somehow subverting the host immune system. maybe this pathogen is causing these genes to actually go down in expression. Alternatively, this may be a way for birds to modulate their immune response. Maybe if they were to turn on every gene, it would cause something disastrous like an autoimmune response, and they don't want that to happen. So we're currently testing these ideas about why these genes are paradoxically going down in expression. Of course, there are a lot of genes that we don't know about in the house finch, novel genes, but nonetheless, using this macroarray approach, we got a nice overview of how gene expression changes upon infection. We move forward in technology, and we're using now an approach called microarrays. And in this approach, we're not using nylon membranes, but we're using these tiny glass slides where we print thousands of genes. And we can monitor the expression of each of these genes using special probes and RNA that we have retrieved from the spleen of these birds. And here's just a picture showing what these microarrays look like. They're printed by these tiny robots that print these very small points in thousands of copies on each slide. And they're a very effective way to monitor thousands of genes across the genome. So in this approach, we're using what's called a common reference design. Basically, in this design, we're going to pool the RNA from all our experimental classes into a single pool. And we're going to compare the gene expression of every individual class to that common reference. Okay? You can see our individual classes here. We've got control birds from Alabama, which haven't been infected. We've got birds 3 days after infection from Alabama, 14 days after infection. We've got similar groups for the Arizona birds. We're going to label the control and the infected birds with different dyes, and we're going to pool all the classes into a single class so that we can basically compare the relative expression of any particular combination we want. It's a useful design if you have a small amount of material. We can't measure gene expression on individual birds, but we want to do comparisons among all the classes in our study. So it's a very useful approach for us. Now, we confirmed all the patterns in this microarray analysis with other sorts of classical approaches. For example, we did northern blots, which are a way of asking about levels of gene expression. This approach has been around for a long, long time and is a very powerful and robust way of measuring gene expression. Here's an example showing HSP90. We're also going to use quantitative PCR. Again, these are simply methods to confirm the results of our microarray study. And of course, confirmation and validation are very important parts of any sort of study in genomics and molecular ecology. Now, let's look at some of the patterns that we see in the different groups of birds that we're looking at. On the y-axis, you can see a measure of the number of genes or the number of clones that are showing changes in expression, number of clones being number of dots on those slides. And on the x-axis, you can see different comparisons. Now, what you can see on the left here is that at baseline, before any birds have been infected, we are actually seeing differences in levels of gene expression between Arizona and Alabama birds. This is really exciting. This means there is geographic variation in gene expression. Just baseline levels, no pathogen involved. We can see that there's lots of genes that are showing differences in expression level. So this is a really interesting aspect of gene expression and geographic variation in the species. Now, when we look at day 3 and day 14 within Arizona, what we're doing here is comparing birds that have been uninfected, that are clean of the pathogen, versus birds that have been infected. You can see that in both day 3 and day 14, quite a lot of genes are showing differences between uninfected and infected birds. That's exciting. That means we've got some information to work with. So we've got a lot of differences between control and infected birds in Arizona. Remember, Arizona birds are never seen the pathogen in nature before when this experiment was done. Now let's look at Alabama birds, which have had about 12 years of exposure to the pathogen at the time this study was done. Notice how on day 3, when we compare day 3 birds with control birds, we actually cannot find any genes that show differences in expression between uninfected and infected birds. This is amazing. Somehow these Alabama birds have figured out how to cope with this pathogen without changing the expression of any genes that we can detect. When we look at day 14, we see a small number of genes, much smaller than what we see in Arizona. So this is really cool. This says that Arizona and Alabama populations are responding differently to experimental infection by mycoplasma. This is just another way to look at some of those data. You can see in the green bars, all of these are levels of expression comparing Arizona to Alabama. So in the top, you can see all these immune system genes, which are showing higher expression levels at baseline. This is before any infections. In the yellow, you can see those same genes between Arizona and Alabama after they've been infected. And what you can see, like we saw in the earlier study, these genes are being suppressed, being downregulated in the Arizona birds. This is a very surprising pattern. Arizona birds showing lower expression compared to controls and compared to Alabama birds. So maybe again the Alabama birds are showing a more robust response. They're not being subverted by the pathogen, perhaps. This shows a long list of all of the genes that show differential expression in our study. Large number of genes belong to the immune system, to metabolic processes, lots of different genes with lots of different functions. What I want you to pay attention to is this green column, number 4, which shows that, again, many genes in Arizona are showing lower levels of expression than are genes in Alabama once they've been infected. This is, again, differential response of these two populations with different histories to experimental infection of this, with this pathogen. Yet another way to look at the data. Never hurts to look at it in many different ways. You can see, again, Arizona versus Alabama, the immune genes tend to be downregulated in Arizona birds. By contrast, stress genes, which you can see on the right there in blue, or in purple I should say, these genes are showing higher levels of expression in Arizona versus Alabama birds. Exactly what you might expect for a naive population that had never seen this disease before. So really intriguing comparison. Now, let's look at the dynamics of change between these three populations. Remember in the macroarray study, we looked at birds using, back in 2001, we looked at the Alabama birds. And in the more recent study, we've looked at birds from Alabama and Arizona in 2007. Let's look at the similarities and differences between all three of these populations. What you can see on the y-axis here, this is the percentage of expression differences as compared with the Alabama birds in 2001, right? So what you see is that Alabama birds in 2007, in this column here, are showing a lot of differences from the same population a mere 6 years earlier, okay? That's really, really interesting. By contrast, the Arizona birds from 2007 show quite a bit more similarity with the Alabama birds back when they had less exposure to this pathogen back in 2001. This is a strong suggestion that the Alabama birds have undergone evolution. They've changed between 2001 and 2007. And that is our current hypothesis that, in fact, Alabama birds have undergone evolutionary change such that their response in 2007 is quite different from the response in 2001. By contrast, Alabama birds in 2007, which had never seen the disease, are much more similar to Alabama birds were in 2001, which also had had minimal exposure. Interesting comparisons among all three of these populations. This just summarizes those results. The signature of microevolution in gene expression appears to have occurred in Alabama birds, as indicated by this dotted line here. The solid line indicates these points of similarity between the Alabama birds in 2001 and the Arizona birds in 2007. And of course, as we saw, the Arizona and Alabama birds in 2007 were quite different from each other in many respects. All of these comparisons suggest that evolutionary change has occurred in the Alabama population and is one suggestion that disease can be a potent driver of evolutionary change in bird populations. Finally, we can look at the functional consequences of these gene expression changes. And so what you see here are two panels showing what we call the mg load. This is simply the number of Mycoplasma bacterial cells per cell of host, okay? So higher bars means higher amounts of disease in the particular bird. You can see that the Arizona birds are sustaining higher levels of mycoplasma than are the Alabama birds. This again suggests that they're not as good at dealing with this pathogen. What you can also see here on the right is that we can develop a simple function of gene expression to show how that can drive the level of mycoplasma down as a simple function of these patterns of gene expression. And what this tells us is that these expression differences that we're measuring actually have functional consequences for the disease. And this is really exciting. It may mean that we've come closer to figuring out what genes are actually involved with the immune response to this pathogen. Okay. Now we're going to end the talk by talking about the other side of the coin, namely the pathogen. Pathogen is this very simple organism, much simpler than the host, but nonetheless, as we'll see, evolving in very interesting ways. The genome of Mycoplasma gallisepticum was sequenced in 2003, and it is less than 1 million base pairs. MB stands for megabases. Now, a million may sound like a lot, but in fact, it's a tiny genome. There are relatively few bacteria that have smaller genomes than that. By comparison, of course, the human genome is 3 billion base pairs, and the House Finch genome is about 1.4 billion base pairs. So this pathogen is a much, much simpler genome, and for that reason, we can really look at the entirety of its evolution in very detail. This is a picture of the pathogen from the poultry sequenced in 2003. And this is a picture of the House Finch genome, which we've recently sequenced. You can see it looks quite similar. It's also circular, like the poultry pathogen. You can see the variations, the differences between the House Finch and the poultry strains are visualized on the outer blue circle. There are lots and lots of differences. Lots and lots of evolutionary changes occurred. On the red circle in the second in from the outside, you can see all the variations that we find among the House Finch strains that we've looked at. So what we did in this study was to pick 12 House Finch strains at different time points during this epizootic. We picked a few from the very early part of the epizootic in the mid-1990s. We picked several from 2001 and several from 2007. We've compared those with some new strains from poultry that we've also sequenced, and we've been able to look at many different aspects of the evolution of this pathogen by looking at these sequence comparisons. And in this slide, you can actually see some of the differences we found. So for example, these gray bars here, like labeled 4 and 5, you can see those are deletions that have occurred in the House Finch strain. It amounts to about 50,000 base pairs that simply are not present in the House Finch Mycoplasma strains. We can also look at these so-called insertion sequence elements, IS elements, which are sort of jumping genes. You can see there are some novel positions of those IS elements in the House Finch strains relative to the poultry strains. Now, let's look at our old friend genetic diversity. You can see genetic diversity on the y-axis, and what you can see on the x are different samples, both from poultry and from house finches at different points in time. The first two columns on the left you can see are chicken and turkey sampled from the mid-1990s, about the time this pathogen jumped to the house finches. They have a lot of genetic diversity. You can see that it's between 0.03 and 0.04. That means about 4 positions in every 100 are different, are variable within those groups. On this scale, you can't even see the genetic diversity of the house finch strains. It's so low. We have to blow it up to a new scale, which you see on the right. And what you can see is that, yes, there is some diversity in the house finch strains, but overall it's about 2% of the diversity that we saw in the poultry strains. And so clear evidence for a founder effect. As this pathogen jumped from poultry into house finches, the genetic diversity was lost because only a few pathogens, a few individual bacterium made the jump successfully. We also see that there's some variation over time in the house finch strains. You can see that this 2007 strain has much lower genetic diversity than does the 1994 or the 2001 strains. So this shows that there's continuing bottlenecks, continuing founder effects within the House Finch Mycoplasma lineage. Let's look at the gene tree of Mycoplasma isolates, okay? And from this, we're going to make some inferences about whether the population is stable or a constant size, or whether it's expanding, growing in size. We expect from population genetic theory to see a gene tree with fairly long internal branches, such as you see here on the left, if the population has been stable, okay? Hasn't been growing or shrinking. If a population has been growing, as you see here on the right, we expect to see a gene tree in which a lot of the coalescent events are crammed into a small temporal window, which it turns out corresponds to the time of population expansion. This is because going backwards in time, it's as if it's going through a bottleneck. And we have a big population here on the present day. All the lineages are coalescing rapidly as the population is contracting going into the past. So we can actually model the time and the extent of population growth from gene trees such as we're going to make from the pathogen. Here's a so-called skyline plot developed with some software called BEAST. B is for Bayesian. We're going to talk about Bayesian statistics again. And you can see that we're going backwards in time. We've got years before present on the x-axis, and we've got a measure of effective population size on the y-axis. You can see that going backwards in time, well, actually, let's talk about going forwards in time. It's probably more intuitive. We actually can see some evidence for a population expansion about 17 years ago. You can see the effective population size is somewhere between 10 to the second and 10 to the third 17 years ago, and then it shoots up to above 10 to the third, or 1,000, by about 14 years ago. This is the signature in the genetic variation that's showing evidence for a population expansion. You can also see a dip in the estimated effective population size. This could be a bottleneck. It could also be a bout of natural selection, which has caused a reduction in genetic diversity because one variant has come and swept to fixation in the population. So very detailed dynamics that we can get from this sort of data. Here is a tree of the bacterial lineages. You can see the lineages from 2007 here at the top. You can see lineages from 2001 in the middle, and then the earlier lineages from the mid-90s below. Couple interesting things to note. First of all, there is a clear clustering by time period. We've seen sort of a sequential movement from the mid-1990s, the early 2000s to 2007. We also see some evidence for intermingling of the poultry and house finch strains early on. This brown lineage, TK2001, is a turkey strain sampled in 2001, and it is closely related to one of the house finch strains sampled from Tennessee in 1996. This might mean that this lineage has actually moved back into poultry from house finches. Maybe we've got some ongoing exchange going on. You can also see that the poultry lineages down at the bottom are really the wellspring from which the house finch diversity arose. So our founder event occurred right at the base of the house finch lineages. Now, here are a couple other interesting aspects of this tree. First of all, we estimate a coalescence time or a point of common ancestry of these mycoplasma lineages a fairly long time ago. We estimate over 600 years for the common ancestor of the house finch and chicken strains. The most basal, the reference strain, which is at the bottom there, called R. low. Clearly, this is way older than the pathogen in the house finches. And what this tells us is that the reference strain, R. low, is not the source of the current house finch mycoplasmas that are circulating today. Some other strain, which is more closely related to the house finch strains, is the source. Perhaps this turkey strain that we mentioned earlier. We also estimate an extraordinarily high substitution rate. Substitution rate is like the mutation rate. It's the number of variants that get incorporated into the population per year or per generation. This measure of 10 to the minus 5 is very, very high even for a bacterium. And this shows us that this pathogen is evolving extremely rapidly, probably trying to adapt to its new host. Finally, we see a lot of turnover, genetic turnover in these successive cohorts. We can see that the 2007 lineage is defined by a number of unique genetic variants indicated by the star here that distinguish it from earlier cohorts in 2001 and 1994. So again, this might be a signature of ongoing evolution, adaptation to this new host. Now, we're going to end by talking not about the host immune system, but about the immune system of the bacterium. This is remarkable. Before I started this study, I didn't really even know that bacteria had an immune system. Turns out they have a set of genes called CRISPR loci, that stands for clustered interspaced short palindromic repeats. These CRISPRs essentially are the bacterial immune system, helping it fight against viruses and phages, things that are even smaller than a bacterium. Turns out that if a bacterium has a healthy variable set of CRISPR loci, it can shut down the proliferation of phage and viruses in its cells. It can essentially turn them off, prevent them from replicating, as you see here on the bottom panel. By contrast, if a cell does not have a healthy, diverse set of CRISPR loci, phage and viruses can proliferate at will and will eventually infect the entire cell and lyse it, namely kill it. And so that's what you see in this upper panel. And suffice it to say that it's adaptive and advantageous for cells to have a lot of these CRISPR loci. And I want you to remember this as we look at what's happened to the CRISPR system in these House Finch lineages of the mycoplasma. This shows a diversity of the CRISPR loci in the House Finch mycoplasma lineages. You can see the 12 House Finch strains that we've looked at from 2007 back to 1994. You can also see, for comparison, a CRISPR loci set in the reference strain of the poultry mycoplasma strain. Now, the poultry strain had over 70 different CRISPR loci, a very large and diverse set. By contrast, the House Finch strains had no more than 52. Now, on top of that, what you can see as we move through time is that House Finch lineages in the mid-90s had a reasonably complete set of CRISPRs. As we move to 2001, they're showing deletions and less diversity. When we finally get to 2007, they're showing incredibly low diversity of their CRISPR repeats. In fact, you can see these black ovals over towards the left of the slide. These are so-called Cas genes. These are called CRISPR-associated sequences. They are essential for the functioning of the CRISPR system. All of the strains from 1994, 5, and 2001 have Cas genes. By contrast, the Cas genes have actually been deleted from the 2007 strains, indicating that their CRISPR system has not only been truncated, but it actually doesn't work at all. So this is really remarkable. We're seeing this pathogen jettison its own immune system as it's moved into this novel pathogen of the House Finch. Why would a pathogen do this? Well, it may be that it's providing a short-term advantage for the CRISPR system and for the pathogen as a whole. It may be that by getting rid of the CRISPR system, it can actually replicate faster. I would predict, however, that because it's gotten rid of this system, in the long term, it's going to catch up with it, and that viruses and phage will be able to infect these lineages more than they would lineages that have an intact CRISPR system. So we've used genomic techniques to look at a variety of questions in this host-pathogen interaction. We've seen evidence for microevolution, very rapid, small-scale evolution of the House Finch in terms of its gene expression when we compare the Arizona and the Alabama populations at different time points. We've seen how the parasite has jumped from poultry into House Finches and has undergone a series of genetic changes, including getting rid of its own immune system as a way of adapting to this new host. Again, a very paradoxical result, but one that may characterize other host pathogen systems as well. Finally, I want to emphasize that because we've been able to look through time at different time points in the system, in the first case looking at museum specimens and using the genetic variation of the House Finches, later on using archived samples of the birds as well as the pathogen, we've been able to look at the details of the genetic dynamics through time. And this, I think, is the real future for looking at these patterns of genetic variation. Combining genomic techniques with well-archived, well-curated, well-documented samples is going to be the key to answering some very exciting questions in molecular ecology and evolution. And I'd just like to thank the large number of very talented people who have helped me in these studies. People working on the House Finch gene expression system, folks working on the pathogen evolution system. In short, science today takes large numbers of collaborations, people and scientists with different scientific skills. So that's what makes science so much fun, is working with people with different skill sets, with different domains of knowledge. So thank you very much.